Well, hello and welcome everyone. Thank you for joining us today. I'm Tracy Smith, the Collaboration, Education, and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. And in this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we'll get to hear from two different speakers who will discuss two different topics. First, we'll focus on bridge integration, a computational method that harmonizes single-cell datasets across diverse molecular modalities by using multi-omic data as a molecular bridge. Then we'll move on to NTT-seq, a new assay that allows for simultaneous profiling of multiple histone modifications in single cells. As always, we'll have time after each talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I will read it aloud to the speaker. So our first guest today is Yuhan Hao, a PhD student at the New York University and a member of the CICA lab at the New York Genome Center. His research has focused on developing new computational tools for analysis of single-cell genomics data, particularly for single-cell multimodal data clustering and cross-modality integration. And our second speaker today, Tim Stuart, is a postdoctoral fellow at the New York Genome Center and develops computational and experimental methods for single-cell epigenomics. Before moving to New York, Tim completed his PhD in biochemistry at the University of Western Australia in the lab of Ryan Lister, where he studied DNA methylation and transposable elements. And with that, Yuhan, I will hand it over to you.
Okay, thanks. Thanks, Tracy, for your introduction. And let me share the slides. Okay. Okay, so hello everyone, I'm Yuhan Hao, the PhD student from Rahul's teacher's lab. I'm very excited to present my work here. Today I'm going to talk about the dictionary learning for integrative multimodal and scalable single-cell analysis. Currently, one of the trends is changing the single-cell genomics, the idea of mapping cells to the reference. As the artists developed more from Fabian's lab showed how you can take a reference dataset and map your query data onto it. And our lab also set up a web app called Asmus. It's kind of like BLAST, but for single cells. Researchers can just upload their count matrices of their data and click one button. Like BLAST, they can get automatic analysis and cell annotations. Right now, we have built a reference for 8 different tissues. This is great, but the problem that is all just for the RNA-seq. In order to map other modalities such as protein, chromatin accessibility, histone modifications, we need to have the reference dataset for all of them individually. If we have to build a series of new reference for all different tissues, every time when we want to map a new modality, it's literally impossible. And in Thera 3, team in our lab did some pioneer work to integrate the single-cell RNA-seq and the single-cell ATAC-seq together. He converted one set of features into another. For example, taking the gene body sum of ATAC-seq signal as a proxy for transcriptional output, the gene activity score. And here, he integrated the single-cell RNA-seq and ATAC-seq data from Morse cortex together. Generally, right, the cells from the same cell type are grouped together in this context. While this transformation, it assumes a strict and a simplistic biological relationship between the modalities that may not hold true, particularly in developing or transitioning systems. But currently, we have many new technologies on single-cell multi-omics, which can measure multiple modalities of single cells simultaneously. And these technologies give us new opportunities to perform integrations across modalities. It helps us translate the information between different modalities just within the same cell. Here, I will introduce our methods called bridge integration that harmonize a single-cell dataset across modalities by leveraging a multi-omic dataset as a molecular bridge. It is a robust integration method without requiring any strict biological assumptions. Apparently here, we don't need to worry about the question if there is a path through all individual modalities that only cross all 7 bridges once that bothered Euler 300 years ago. But currently, we only need to focus on 2 datasets and 1 bridge in the middle. And this is what we are going to address with our dictionary learning. First, let me introduce what dictionary learning is. It is a representation learning commonly used in the image analysis, but not really in genomics. The idea is that you have a dataset, like a noisy image, then you take a subset of pixels from the input. So in this case, they are the little patches from the image. And then you have to reconstruct the whole image as a weighted sum of dictionary elements. And if you do this, the dimension of the image is reduced from millions of pixels to hundreds of little patches in the middle here. You can get a relatively low dimensional representations and can easily reconstruct a denoised image. But the key thing is that you are learning how to represent a dataset as a combination of individual elements. And that collection of individual elements is called dictionary. Obviously, the dictionary is important. It turns out that you can learn the dictionary just by taking the patches from the data itself, the noisy image. However, you can also take patches from a different dataset, say a collection of separate but high quality image. Both methods can work well. Next step is that how we can use the dictionary learning to solve our question. Our question is that how we can map dataset when they measure different things. Here's the example on the left. We have built a fantastic RNA reference for human bone marrow, especially for the early differentiating progenitors. Now, on the right side, we have the ATAC-seq that is from Greenleaf's lab. We want to annotate those cells, but we cannot use our reference because it's built on genes. ATAC-seq measures the peaks. So what do we do? The intuition is that we're going to make use of a third multi-omic dataset, which measures the RNA and ATAC as a bridge to map the ATAC query to the RNA reference. And how can we do that? And this is where dictionary learning comes in. Here, each RNA cells on the left can be represented as a weighted combination of dictionary elements. Because the multi-omic cells measures the RNA, and we can do that. And each ATAC cell on the right can also be represented as a weighted combination of dictionary elements, because the multi-omic cells measures the peaks as well. So the trick is that both single-cell RNA-seq and ATAC-seq dataset can be represented by the same dictionary. So these 2 datasets can be compared and integrated in the same space, a multi-omic cell space. So this is our ATAC-seq mapping result. On the left is our Asmus RNA-seq reference. On the right is a mapped ATAC-seq dataset. All work we did to annotate and refine the RNA-seq reference can help us interpret new ATAC-seq data as well. It just opens the door to a new modalities. Now, I will highlight one cool result. If you look closely here, you can see that I have circled a group of cells in the ATAC-seq dataset that we annotate as ASDC, some of the rarest cells in this dataset. This is actually a population that Rahul discovered in his postdoc. It is a new type of dendritic cells, which was originally discovered by single-cell RNA-seq. It's about 0.04% of cells in the blood and the bone marrow. ASDC was defined by 2 markers, AXL and cyclic-6 genes. And here we found those cells in the single-cell ATAC-seq dataset. When we look at the marker gene, cyclic-6 locus in the ATAC-seq data, here is a unique enhancer that is open only in this ASDC. Those cells would have been impossible to find in the single-cell ATAC-seq without this bridge integration. From the above the results, you can see that the bridge integration works very well, but what are the requirements for the bridge dataset? To answer this question, we did some benchmark analysis and ground truth using the ground truth dataset. So the conclusion is that the bridge does need to be representative, but it doesn't need to be very big. So if you just have 6 or 60 cells from each cell type in the bridge dataset, you're good. You can get very high levels of accuracy, and you can get away with even less. Even if you have around 20 or less, you can still get high accuracy for some cell types, but there are some risks. And in some cases, you will fail to accurately map extremely rare subpopulations. But I think this is a good guide for experimental design, and 50 or 60 cells per cell type is not a lot. Even the bridge dataset only have a few thousand cells can be very helpful. And I give you an example of RNA and ATAC integration. But our bridge integration is a general strategy for cross-modality integration. The middle box here listed alternative multi-omic technologies that can be used as a bridge dataset. Just as another example, we took a dataset generated by Chongyuan from Akers lab, where they measure the DNA methylations in single cells from the human brain. But actually, DNA methylation is hard to annotate. So a lot of clusters are just labeled as -1 or -2. It's just hard to know what those clusters correspond biologically. And earlier last year with Allen Brain Atlas, we released an ASMA's RNA reference for the human cortex, annotated with a curated cell ontologies. Here, we use a multi-omic SNTT-seq as a bridge to connect the RNA reference and DNA methylations. We can map DNA methylation cells to our RNA reference. And here, this is the annotated DNA methylation cells. We don't really discover a new cell type or anything novel, but we can label things with the defined ontologies. So we don't need to call the cell cluster 1 or cluster 2. We can automatically give them annotated things like layer 6 IT or layer 6 CT. Okay, so this is about the bridge integration. But as we see, the bridge integration results all assume that we have a comprehensive and high quality reference. In the second part, I will show you that one recent challenge about the reference building. So the success of technology development has led to exponential growth of skills in single-cell RNA-seq data from tens of cells up to hundreds of thousands of cells in around 1 decade. In particularly, two recent technologies, scRNA-seq 3 and CyTOF open up a new area of millions of cells. Unavoidably, the expansion of data gave us more questions. With this large-scale data most commonly used, computational methods become infeasible, either requiring huge memory capacity or take a very long time to run. To take the benefits of these high-throughput technologies, we utilize the dictionary learning to analyze this large-scale single-cell dataset. As I mentioned earlier, dictionary elements can be learned within or across dataset. The bridge integration is based on cross dataset dictionary. We also build a within dataset dictionary learning to do the community-wide dataset integration. The key method is that we sample the representative cells from each dataset as dictionary, and we call those sampled cells as atoms. And then we can build a sampled cell representations for each individual dataset. But the first question is that how can we get the representative cells? The key idea we used is to sample cells based on the Levy score, which measures the importance of each individual cells. We computed the Levy score based on the methods proposed by Clarkson and Woodruff. It is a fast approximation of the Levy score by using random sketching and the random projection. Instead of explaining those matrices here, I will show you some applications of the Levy score. To begin with, I calculate the Levy score for a single-cell RNA-seq dataset from tags, which profile 33,000 PBMCs. So the UMAP is colored by their cell type, and there are both abundant and rare populations in this dataset. And we observe that the smaller population like HSC, plasmablasts, and PTC indeed have bigger Levy score. And the abundant populations such as those naive T cells have smaller Levy score. Then I average a Levy score within each cell type and to study the relation between the population size and the Levy score. So here, each dot on the right plot represents one cell type. And the x-axis is the number of cells for this one population. And the y-axis is the averaged Levy score. Interestingly, we observe an inversely proportional relation between the population size and the Levy score. Here, I need to emphasize that when we calculate the Levy score, we know nothing about the cell type information. We only used the sparse gene expression matrix. In this case, when we sample the representative cells based on the Levy score, we can make sure that all small population will be selected and the big population will not be lost as well. So based on the Levy score, I sampled 5,000 cells from 33,000 cells and reanalyzed them. The right UMAP is from those 5,000 PBMCs. All rare populations are maintained, like HSC, PDC, plasma blast. All major cell types are preserved as well. So usually you would never expect to observe so many cellular diversity within 5,000 PBMC. And this dataset is not a real big dataset. Let's try Levy score on the mouse embryo atlas. So this is a dataset contains 2 million embryo cells from stage E9.5 to E13.5. It only takes around 10 minutes to calculate the Levy score for over 2 million cells. On the contrary, performing PCA for this dataset will cost over an hour. Still, we observe the Levy score is inversely proportional to the population size. And here, we have to sample the cells based on the Levy score, right, instead of sorting the cells. And the goal of sampling based on the Levy score is to make the populations become more balanced. Okay, I assume that I have convinced you that we can get representative cells from the dataset and use them as a dictionary. And how is this related to the integration? Let's look at an analogous story. I take a toy example. We have an article and a dictionary. Right? We can construct a dictionary index for each word in the article. And based on the dictionary index and dictionary themselves, we can fully reconstruct the article as well. So the basic elements in the dictionary are the words. Okay, so here, if we want to convert the American English words in the article to the British English spelling, we can directly translate the article. But when we need to translate millions of the articles, it will become inefficient. But under this big dataset context, it will be more efficient to translate the dictionary from American English to British English, and then use the same dictionary index to indirectly translate millions of the articles. Okay, so the same ideas can be used in the single cell integration, RNA integration. Here, we integrate those sampled cells, those items or dictionaries to get rid of the batch effects. And then use a batch corrected dictionary and the dictionary representations built in the RNA assay to reconstruct the batch corrected expression profile for all the cells. And we call this atomic sketch integration. So here we collected over 1.5 million of long cells across 19 public projects. This is a UMAP visualization from original RNA expression. The batch effects are very strong. Cells are not only grouped by their biological difference, but also grouped by their sequencing batches. Then apply the atomic sketch integration. So this is a batch corrected UMAP visualization. Different batches are mixed evenly, and it only takes 55 minutes to complete the whole process using one CPU core. Without sketching, it takes over 24 hours to integrate all this data set. I want to highlight an ultra-rare population, the alnocytes, only 0.03% among these 1.5 million cells. The population express many cystic fibrosis genes. Those are the genes that have mutations in the cystic fibrosis patients. Due to the small number of those cells, the population was largely ignored before. In our integration results, the population is clearly grouped together. And in the sketch integration, I only sampled 5,000 cells from each batch. But based on the compositional ratio, there will be only 1.5 alnocytes if the 5,000 cells are uniformly sampled. However, here we sampled each dataset by Levy score. So you can see that the alnocytes have very high Levy score value to guarantee that they will be existed in our dictionary. Okay, so here is the last example, combining both within and across dictionary learning. We built a large-scale PBMC RNA reference atlas using our atomic sketch integration methods to integrate 3.4 million of PBMCs from 274 individuals. Then we took a CyTOF dataset from Combat, a UK consortium, where they measured 47 surface and intracellular proteins of 5 million blood cells from COVID samples. Using the RNA reference just built on the left and the PBMC CyTOF as a bridge, we can easily map the CyTOF data to our reference and get comparable cell type annotations. To validate if we transfer correct annotations, we generate a heat map showing the reproducible intracellular and surface protein markers of cell types that are consistent across 200 individuals. So each column here just represents a pseudo bulk average of all cells from one cell type and a single individual. Here I highlighted 3 cell types with intracellular markers or hot annotated. First is the Tregs. You can see it express high level of transcriptional factor FOXC3 across most individuals. And the mate cells were uniquely depleted for the expression of the cytotoxic granzyme B, consistent with previous reports. And finally, we successfully annotated a rare population of the innate lymphoid cells, which were not independently identified in the CyTOF dataset, but it correctly exhibited a CD161 positive, CD25 positive, and a CD56 negative immunophenotype. And taken together, we demonstrate that how bridge integration can help us interpret and improve the resolution of cell type annotation, including the extremely rare cell types. And the atomic sketch integration can help us build a very large-scale community-wide big RNA reference. And both methods, they can facilitate researchers to annotate their new query dataset. And the curated annotations in the reference can also can transfer them to the query dataset. Okay, and our manuscript is submitted to the bioRxiv, and you can also find the corresponding variants in our website, one for the bridge integration, and we'll have another one for the atomic sketch integration. Okay, so in the acknowledgement, first I have to thank Rahul for his insightful ideas and guidance to make this project possible. And thanks for team that he made many contributions on the early stage of this work. And thanks for all other lab members for their endless help and encouragement. So, and lastly, thank you for your listening. Okay.
Well, thank you, Yuhan. That was a really great talk.
Thanks.
And we have some time to take some questions now. And you can raise your hand or type your questions into the chat. And here we have a first question. So how does your dictionary-based method to build the reference atlas compare to SCRNA-seq?
Okay. It's an interesting question, right? As we know, right, SCRNA-seq, they use deep learning model to generate, to build the reference and map the query onto it. But the purpose of the dictionary learning, the atomic sketch integration, is mainly designed for the large scale data set. In all the integration process, actually, right, in this integration, actually, even though you don't have a large memory capacity, it's kind of totally fine because we never loaded all the data sets simultaneously into the memory. We can do them, we can sketch those representative cells data set by data set and just have a small subset of those dictionary, those atoms. Those are some of a small data set. And then you only need to do the integration of those cells instead of the entire data set. And the SCRNA-seq, right, when they use the deep learning models, yes, they kind of split the data set by the batches, but they kind of use it, they not really emphasize those small rare populations. And we think that this library score-based methods can kind of make sure that those small rare populations will not be lost and also require kind of less memory.
Okay, thank you. Another question we have, how does this dictionary-based method perform on the pairs of data sets from different tissue sources? Example being annotating the tumor microenvironment immune cells based on a reference of PBMCs?
Yes, that's interesting questions, right? Actually, that is mainly depends on your integration methods. In our dictionary-based methods, we mainly just this is general framework for after integration. Then you can use the integrated cells, right, to reconstruct your original data set. So really depends on if your integration methods, right, they kind of can handle those tissues difference, they put the immune cells from different tissues together, then yes, the dictionary learning this stuff definitely can accelerate that process. But if you use different integration methods that they would consider the tissue difference as kind of true biological methods, then right, the dictionary learning this methods this way can still maintain those tissue stuff, right? So if it's can work for different tissues, the immune cells from different tissues, it mainly depends on what integration methods you used. And the dictionary way here just to accelerate the integration process.
Well, if we don't have any more questions right now, I thank you again, Yuhan.
Thank you.
And, next, we will hear from Tim Stuart. Tim, feel free to take it away.
Okay, great. Thanks, Tracy. Let me just share my screen. Okay, so yeah, today I want to talk about a new method that we've developed called NTT-seq that enables measuring multiple aspects of chromatin state at single cell resolution. And just to give a little background on first why we're interested in chromatin states. Basically, as I'm sure many of you know, these chromatin states are extremely important in defining cellular phenotypic heterogeneity because each cell has essentially the same copy of the genome but interprets it in different ways. And from many decades of research, we also know that these chromatin states are extremely important in development and in certain types of disease. And an interesting point here is that the chromatin state is sort of inherently combinatorial. So it involves many different things, including histone post-translational modification, transcription factor binding, RNA polymerase binding and activity, insulators, CTCF, DNA accessibility, and all these different factors, right? So these all come together to, at a specific locus, define really the functional activity of that chromatin. And so we really need to be able to observe all of these different things, or at least multiple aspects of chromatin state at once, to be able to fully understand how the chromatin is functioning. And that was the goal in our work here, is to basically develop methods that will enable us to do that, and with single cell resolution. So there's been a lot of work done so far to measure individual aspects of chromatin state with single cell resolution. So there's assays like single cell ATAC-seq, which is now very popular, and more recent methods like CUT&Tag, developed by Steve Henikoff's lab a few years ago, and other very similar methods as well. And these methods are able to measure basically one aspect of chromatin state, like a histone modification genome-wide in single cells. And the way that they do this is a little bit similar to, at least conceptually, to ATAC-seq, where we have a TN5 transposase being added to the chromatin and tagmenting DNA and incorporating these sequencing adapters that allow us to then amplify and sequence those fragments of DNA. But the key idea in CUT&Tag and the other sort of related methods is that we can use an antibody to guide TN5 to certain regions of the genome that we're interested in. So here we can stain the chromatin with an antibody that might bind to a histone modification, for example. We can then add this protein A TN5 fusion protein, and here the protein A will bind strongly to the antibody and effectively tether TN5 enzyme around regions of the genome where the antibody is bound to. And then we can activate the TN5 and cause the insertion of those sequencing adapters specifically around regions where the antibody is bound. And obviously we can sequence that and then learn, where that antibody was bound across the whole genome. So in this work, what we were interested in doing is essentially taking this idea, but developing a way that we can multiplex the antibodies that we're adding, so we can add more than one antibody at a time and measure, more than one different histone modification or transcription factor or different things in a single experiment. And the way that we did this is we looked at this a different innovation known as a single domain antibody, or also known as a nanobody. And these are interesting types of antibodies produced by some animals like sharks and camels and alpacas that contain only the heavy chain part of the antibody. So if you look at the mammalian IgG antibodies, which you might be familiar with, they contain the heavy chain and the light chain. And so the part that binds to the epitope is made up of two different protein sequences. And so this makes it a little bit hard to engineer this into like a smaller protein for more biotechnology applications, because the binding site is split across these two different proteins. But that's not the case in these other types of antibodies where we just have this single chain. And so in the camel and alpaca antibodies, for example, we could just take the tip of the antibody, this heavy chain part, and this would define a protein that will bind specifically to an antibody, to an epitope, and be very small. So our approach here is to basically engineer a new set of nanobody TN5 fusion proteins, and we were very lucky in that a few years ago, in this paper by Pliner et al., they actually described the sequence of 4 different nanobody proteins that will bind specifically to different types of IgG antibodies. So we took these protein sequences, coupled them with TN5 in a fusion protein, and produced these. And so now we have these 4 different nanobody TN5 fusion proteins, and they recognize different types of primary antibodies. So we have 2 species-specific ones. One will recognize mouse antibodies, the other rabbit, and then 2 that would recognize specific mouse IgG isotypes, so IgG1 and IgG2A. So the first experiment that we did here is just to test, whether these do indeed, target TN5 to a specific primary antibody that's bound to the genome. So we did a few different experiments, staining chromatin with a primary antibody for K27 trimethylation. And we varied whether this was a rabbit or a mouse or a different mouse isotype primary antibodies. And then if we add the different nanobody TN5 fusion proteins, we can observe, whether or not we get tagmentation of the genome at the site. So basically what we find is that only when we add the nanobody TN5 that matches the primary antibody can we tagment DNA and amplify it. So here we have the rabbit primary antibody and the rabbit nanobody TN5 added, and we amplify DNA as we would expect. But when we add the mouse nanobody with the rabbit primary antibody, we amplify basically nothing. So this indicates that that indeed the nanobody TN5 is being tethered specifically to a certain antibody that we're interested in. And this is consistent across the different nanobody TN5 fusion proteins that we have. So this is very encouraging, and what we wanted to do then is convert this into a sequencing-based assay. So we developed this assay called NTT-seq for nanobody tethered transposition followed by sequencing. And the way that this works is very similar to CUT&Tag, where we take cells or nuclei, we can then stain them with antibodies, and in this case, rather than using a single antibody, we can stain with a cocktail of antibodies for the different things that we're interested in. And then we can bind our corresponding cocktail of nanobody TN5 fusion proteins. And the key point here is that we can load the nanobody TN5 fusions with barcoded adapters. So then when we sequence the DNA that comes from this experiment, we can also sequence a short barcode sequence that tells us what nanobody TN5 the DNA fragment originated from, and obviously then what primary antibody was bound at that site. So to test this, we did a bulk cell experiment to begin with, using K562 cells. And here we looked at 3 different chromatin marks. So we looked at the binding of RNA polymerase 2. This is the serine 2,5 phosphorylated version, which is basically actively transcribing RNA pol 2. We looked at H3K27 trimethylation, which is a repressive histone mark, and H3K27 acetylation, which is a mark found in active enhancers. And in the first 3 tracks here, these are from a multiplexed experiment where we put in all of these different antibodies at once and our cocktail of nanobody TN5 fusions, and then sort of demultiplex everything in silico after sequencing. And then in the bottom 3 tracks, we have basically the same thing, but done in 3 separate experiments. So we profiled only RNA pol 2, only K27 trimethylation, and only K27 acetylation. And you can see, at least visually by eye, these look extremely similar. There doesn't seem to be any real difference between the multiplexed versus non-multiplexed data. And we can, of course, look at that sort of more broadly across the whole genome, not just at one locus, and as is shown in these heat maps. So if we look at the K27 acetylation peaks, we can look at the amount of signal for each of the 3 different marks in those peaks for the multiplex versus non-multiplexed experiments, and they look very similar. And the same is true basically if you look at the H3K27 trimethylation peaks, and these are peaks from ENCODE. So this was very encouraging, and what we wanted to do next is, of course, convert this into a single cell assay. So this was actually very easy to do because we can run essentially the same workflow that I just described, where we tagment the cells in bulk up front. But then instead of just extracting the DNA, what we can do is take those cells or nuclei and feed them into the 10x instrument and encapsulate all those cells into a droplet. And in that droplet, they then have another barcode incorporated, which is the cell barcode. So now we can sequence, we get the cell barcode, the TN5 barcode, and the genomic DNA sequence. So we did, again, a similar test experiment using K562 cells, but this time we also mixed in HEC cells just to verify that we can, of course, separate the HEC and the K562 cells in silico after doing the single cell sequencing, which indeed we can. And again, we can look at the different tracks, and we can see that the multiplexed single cell data for the 3 different marks looks very similar to the sort of non-multiplexed bulk cell data that we have from the previous K562 cell experiment. So this was encouraging, but again, it's good to look at this more sort of comprehensively genome-wide, and this is what we did here. And just to walk through what all of this means is basically we can quantify in these 3 different peak sets that we have, for 1 peak set for each mark, the amount of signal for that mark in the peak. And so if we look at the K27 trimethylation signal versus acetylation signal in these peaks, we can see that they basically don't co-occur, which makes a lot of sense. That's what we know about these marks. And this is the bulk experiment, non-multiplex versus the non-multiplex bulk experiment for the other mark. But we can look at single cell multiplex versus bulk non-multiplex, and we see much the same story. We can also look at single cell multiplex repressive mark versus single cell multiplex active mark. And again, very similar. There's a little bit more noise in the single cell measurement, but that's to be expected. And then we can also look at genome-wide the single cell multiplexed signal for K27 trimethylation versus the bulk non-multiplex signal for the same mark, and see that it's highly correlated, which is again what we would expect. And we can look at this for the other marks also, the other combinations of marks, RNA pol2 versus acetylation. These are both kind of active marks, and so they co-occur more in the genome, which is expected. And then the repressive mark and RNA pol2, and they are more mutually exclusive. Okay, so then, this seemed to be working quite well, and what we wanted to do next is see if we can sort of couple this technology with another technology that was developed recently at the Innovation Lab at the New York Genome Center, which is this method called ATAC-seq, which some of you may know, and it's a very interesting method that enables you to measure cell surface protein expression alongside DNA accessibility, measured by ATAC-seq, and was published last year. And then, also recent work in our lab led by Bingxi Zhang and Avi Srivastava have sort of adapted the same idea to measure CUT&Tag profiles, so individual histone marks, for example, alongside cell surface proteins, using the same idea. And so what we wanted to try is coupling NTT-seq with cell surface protein expression using this technology as well. And so we profiled around 4,800 human PBMCs. So these are circulating immune cells. These are actually mobilized PBMCs, so they contain some immune progenitor cells that you don't typically see. And we profiled K27 trimethylation and K27 acetylation, and a panel of 173 different cell surface proteins that are important for the immune system. And so we can see all the different cell types that we would expect to see in the immune system, B and T cells and monocytes and so on. And we can also look at the measured protein expression levels in these cells, and we see the expected patterns. So CD14 is expressed in monocytes, CD19 in B cells. We have sort of mutually exclusive expression of CD4 and CD8 protein in the T cells, and so on. And so, this was very encouraging, and I think is a very powerful method to study the immune system by coupling these multimodal chromatin measurements with measurements of cell surface proteins to help sort of interpret the data. So as a sort of final demonstration here, what we wanted to do is apply this technology to a more dynamic cell developmental process to really leverage the single cell nature of the data. So we profiled around 6,000 cells of the human bone marrow where immune development is happening. And again, looked at the repressive K27 trimethylation mark and the active K27 acetylation mark. And you can see these UMAP representations derived from each assay independently here. We have a little bit more resolution to see the different cell types using the K27 trimethylation data. And then we can construct, using the weighted nearest neighbors method, one UMAP that sort of represents both assays shown here. And you can again see all the different cell types that we're able to resolve. And we're particularly interested in this B cell lineage as a sort of example of where cells are sort of dynamically changing from the stem and progenitor cells through to more of a mature B cell state. So what we did is basically subset out these cells, and we can order them on a pseudo-time trajectory using molecule 3. And this pseudo-time ordering reconstructed the expected sort of ordering of cells, where we have the stem and progenitor cells through to the common lymphoid progenitors, the pre-B cells, and then more of a mature B cell states towards the end of the trajectory. And what we wanted to do then is find regions of the genome where the chromatin state changes as you move and progress through from the stem cell state to the mature B cell state. And we found many hundreds of regions of the genome that undergo these changes. And it was interesting to see also that there seemed to be, in many cases, sort of opposing gain and loss of active or repressive marks. So we have clear regions of the genome, such as these shown at the top, where the cells lose K27 trimethylation as they mature along the B cell trajectory, and they sort of correspondingly gain the active K27 acetylation mark. And then we have the opposite also happening, where the cells lose their K27 acetylation mark, and they gain the repressive chromatin state through acquiring K27 trimethylation. So just to sort of try to validate this a little bit using external data, we also looked at the single-cell RNA-seq dataset published a few years ago from Will Greenleaf's lab, also for the bone marrow. And if we look at, for each of these different regions of the genome that we grouped into either the activated or sort of repressed regions, we can look at what gene is closest to these regions of the genome, and how the expression of that gene changes as you go from the HSC to the more mature B cell states in the single cell RNA-seq dataset. And what we found is that these genes that are near the regions of chromatin that are activated become more highly expressed as you move along that trajectory, and the genes that are close to this repressed chromatin state become more lowly expressed as you move from the stem to the more mature B cell state. Again, supporting the validity of these single cell chromatin measurements that we made. So just to summarize, we've developed this new method called NTT-seq that uses nanobodies to guide TN5 to specific primary antibodies. This means that we can multiplex primary antibodies and measure multiple different chromatin modalities with single cell resolution. This method is also compatible with measuring cell surface proteins as well, and we can see very nice single cell protein expression profiles sort of laid on top of these chromatin states. And then finally, NTT-seq can be a powerful method to study these dynamic cell processes that occur as cells are developing, and understand how chromatin states change as the cells differentiate. And just before I finish, I want to highlight some related work that came out on bioRxiv at the same time as our work by Marek Bartosowicz and Gonzalo Costello-Branco. And they had, incredibly, they had the same idea, where they took the nanobody, these nanobodies, and fused them to TN5 to create the same sort of TN5 nanobody fusions. And they also had this really interesting idea of including just free TN5 by itself with a different barcode on it to measure ATAC-seq profiles in the same experiment. And so they had a bit of a different focus on, they looked at the mouse brain, whereas we looked at the immune system. But they showed some really nice data where they have single cell ATAC-seq tracks alongside K27 acetylation and K27 trimethylation in the mouse brain. So again, very, very encouraging and nice to see that the same sort of ideas and very similar methods are obviously quite robust. They work in very different tissues and in different labs. And so if you're interested in this, please also check out their paper. And so with that, I'd just like to thank everybody involved, most especially Ivan Raimondi, who is the inventor of NTT-seq and did all the experiments shown here, and has been a really amazing person to work with on this. Also Stephanie Howe and Silas Maniatis from the Technology Innovation Lab at the Genome Center for all of their help. Bingji Zhang and Rahul Satija from our lab, and also Levan and Dan who are at the Genome Center and at Weill Cornell for their help and collaboration. And also just want to highlight that these plasmids, nanobody TN5 fusion plasmids, are now on AdGene. So if you're interested in running this, you can get them off AdGene. Maybe just take a screenshot or just search NTT-seq on AdGene and you'll find it. So with that, thank you so much, and I'll be very happy to answer questions.
Great. Thank you, Tim. Incredible talk. Thank you so much. And exciting news that the plasmids are on AdGene. So there is a question in the chat here of why the K27 methylation and K27 acetylation show different patterns in continuous versus binary in several figures.
I'm not quite sure I understand the question. Maybe I can go back a little bit.
And feel free to clarify your question if.
Okay, can you hear me?
Yeah.
In this figure, you will see the H3K27 trimethylation showed a continued pattern across the time. But you will see the acetylation show the binary pattern, it looks like. I'm not sure the biological mechanism behind or just the method.
I see.
Problem.
Yeah. Sure. Yeah. Right. Yeah. Good question. I think so. I guess you're referring to this where you have a kind of line here where the state seems to kind of flip more rather than here you have more of a continuous change. I'm not sure, to be honest. But I would suspect that this is more of a limitation of the data rather than something biologically that's happening. I think that in this case, the data that we have is still quite sparse, and the cells, we don't have a ton of cells yet. So I think, with those sort of constraints, it makes it just sort of appear a little bit more binary in nature, especially where, some of these more intermediate states are not well captured. So I think scaling this up to measure more cells and try to improve the sensitivity would certainly help, and would probably, I don't know, but my guess would be it would result in more of a continuous change that you would see.
Great. So beyond, so you showed us cell surface protein expression modalities. Are there other modalities you think that could work, or do you envision measuring intracellular proteins to be something in the future? Or?
Yeah, that's a great question. I think, intracellular would be great. It's can be very challenging. I know a lot of people have been working on that for a while. But some of the things that we're interested in is sort of coupling this with RNA measurement as well. We're thinking about ways to do that. But yeah, certainly layering on different modalities I think could be very useful. I think one of the motivations in measuring RNA pol 2 binding in some of the initial experiments was that we could kind of use that as a proxy for gene expression. But even better if we can measure RNA itself. So that would certainly be very useful.
Thank you. And I think we have another question here. Have you tried mapping transcription factors or chromatin modifying enzymes? Presumably these are less robust primary antibodies or targets.
Yeah, so that's something that we obviously want to do, but is a little bit difficult because we have to use very high salt in the tagmentation here. So this is also true for CUT&Tag and all these sort of antibody-directed tagmentation assays. Currently, you need to use very high salt. So we use 300 millimolar salt, and that prevents TN5 from tagmenting the open chromatin. But what it likely also does is sort of displace things that bind to the DNA more weakly, like transcription factors. And so if we want to profile TFs, it's a bit of an issue. So I think that's another thing that we want to work on in the future, is thinking about how we can either not use high salt or other ways that we might be able to get around that.
Okay, well, great answer. Very exciting directions that that this technology can go. So with that, I'd like to thank you both, Yuhan and Tim, for your talks, and let you all know that this seminar has been recorded and will be available soon on YouTube and on our website, multimodalintegration.org. You can also follow us on Twitter at segs_ICA, or check our website for the latest announcements regarding upcoming talks and events. And with that, I would like to thank everyone for joining, and we hope to see you at our next seminar. So thank you. Have a wonderful day.